Hi, and welcome to the first part of the tutorial series on HIDE-Deconv. HIDE-Deconv is an interactive command line tool for hierarchical cell type deconvolution and analysis of bulk RNA sequencing data. In the first part of this tutorial, I will show you how to install HIDE-Deconv and use it to analyze breast cancer samples downloaded from TCGA. Therefore, I've already created a folder on my computer, downloaded the necessary files, and started a command line in it. To install HIDE-Deconv, we first need to set up a Python virtual environment, which can be done by typing in python 3-m venv.venv, wait until the command is finished, and then activate this command, the virtual environment by typing in source venv bin activate. After that, we need to install HIDE-Deconv, which can be done by pip install HIDE-Deconv, and wait until everything has downloaded. While this downloads, I will briefly walk you through all the necessary files for the deconvolution. First, we have our breast.h5ad, which is a single cell measurement of the human breast downloaded from the Disco Immune Cell Atlas. The other two files are the bulk RNA sequencing data and the connected clinical meta information. The bulk RNA sequencing data, in this case, needs to have each measurement as a single column, with the column name being the sample ID and the row names being the gene names or sample IDs. You should, in the best case, use raw count data. The clinical meta information is structured as a table where we have, for example, T and M classification, vital status, age at diagnosis, and so on. Additionally, we need a column with the sample ID connecting the bulk RNA sequencing data with the corresponding bulk metadata. HIDE-Deconv has now downloaded, and we can run HIDE-Deconv by typing in HIDE-Deconv run and wait until we are asked for further information. This might take a while as there are a lot of packages that need to be loaded in the background. HIDE-Deconv asks us now if we want to create a HIDE-Deconv project structure at the given project path, which we want. So we press in Y, press enter, and enter all of the necessary data. First, we need to enter the path to the undata single cell file, which is our breast.h5ad. HIDE-Deconv now loads the data and asks us to select the column of the cell type annotation. In this case, the cell type annotation is stored at the CT observation, so we select it and press enter. If you want further hierarchy levels, you can press Y now and add all of these levels, but for the sake of this tutorial, we only use the subcellular annotation. Next, HIDE-Deconv needs the bulk RNA expression file, which is the tcga-brca.csv. When we press enter, it will load all the data in it and give us an information on the shared gene set. We can see that between the single cell data file and the bulk RNA sequencing data file, 19,893 genes are the same. For this example, we only want to use 1,000 genes for the deconvolution, 5,000 simulated training bulks, and we want to mix up 100 single cells to create a simulated bulk. After we finished typing in the parameters, we can run the pre-processing and wait until everything is set up. On the left side of the screen, you can see that HIDE-Deconv created some folders, which will be subsequently filled by HIDE-Deconv.
After the pre-processing has been finished, we can train the model by just typing in Y and wait until everything is done. This might take a while depending on the size of your data set, on the chosen parameters, and of course on the number of iterations you chose for training. After the model is done with training, we can choose a deconvolution model and deconvolve our selected data. As to the current state, there is only the HIDE model included, so we have to choose this. Now while this runs, we can have a look at all of the information that was created. We can have a look at the training loss, which can be found under the figures folder. We see our model converged to some point. We can see that we used 1000 genes, 5000 bulks. We only had one cell type layer, and we used 100 cells per bulk for training. Additionally, we get a plot of all the training results where we can compare how good the model was able to estimate a cell type on the training data set. The deconvolved data is then stored under the results folder and the hide folder, where we get the C_sub file for the lowest cell type hierarchy level. All higher cell type layers would have a corresponding name. And we can see in the column names, we find the TCGA sample ID, and on the left side as row names, the cell type names.
As last part of this tutorial, I want to show you how to use HIDE-Deconv for post-deconvolution analysis. All of the post-deconvolution analysis commands can be found under HIDE-Deconv analyze. In our case, we want to use a partial least square discriminant analysis, so we have to type in plsda. If you're interested in other post-deconvolution analysis methods, I would like to guide you to the HIDE-Deconv GitHub repository, where you can find a user guide under docs, where every single command available in HIDE-Deconv is shown and shortly described. So, for example, we find the partial least square discriminant analysis under chapter 4.6. To plot it, we first have to choose our results folder and the cell type layer. In our case, we only use the subcell layer. The next step consists of selecting the sample sheet, which holds the clinical meta information. Then we first have to select the column that holds the sample IDs, so the column that links the measured bulk RNA expression data with the clinical meta information. And then we additionally need one column that splits our data into cohorts. We are interested in the cellular differences between various breast cancer subtypes, so we select subtype. And after some time, you can see that a subfolder has been created here, and you can open the PLS-DA for our data. Additionally, you find a biplot, which not only contains the single data points, but also the areas of arrows of direction of cell types. One last command I want to show you is the HIDE-Deconv help command that can be accessed by just typing in HIDE-Deconv help. The help command will give you an overview over all available commands in the current HIDE-Deconv version, and also gives you further information on how the training data and the bulk deconvolution data has to look like. You can see, for example, all the available analysis commands. You can see further commands for pre-processing and visualizing bulk data, or commands for working with the undata format.